Biological molecules, carbohydrates. Carbohydrates are made up of smaller units called monosaccharides. So the monomer of a carbohydrate is a monosaccharide, and the most common ones that you come across are glucose, fructose, and galactose. All of these are isomers of itself, and I wanted to just show you the functional groups that make up your carbohydrates. You'll see they mostly have hydroxyls, circled in blue, and they have carbonyls, and the carbonyl either is at the end or it could be within the sugar, and that is the one that gives the differences between your glucose, fructose, and galactose. Let's look at that basic structure of glucose in its ring form. Notice in the structure we don't write all the carbons or the hydrogens. We just remember that carbon always makes 4 bonds. So if you had to stop and count, you would see how many carbons are in glucose, how many hydrogen, or how many oxygen. So pause the video and figure it out. Here you can see whether you were correct or not. 6 carbons, 12 hydrogens, and 6 oxygen. So C6H12O6, the structure of glucose. Other important monosaccharides are your riboses. Riboses are 5 carbon molecules important in your nucleotides, DNA and RNA. They both have 5 carbons, and if you have a look at the big difference is that on carbon number 2 in DNA there is no hydroxyl group, whereas in RNA, the ribose sugar, there is still a hydroxyl group. Monosaccharides are joined together to form disaccharides. The bond is known as a glycosidic linkage, and this is due to dehydration synthesis. So here you can see a molecule of glucose plus fructose forming sucrose. Polysaccharides are when we start linking glucose together by dehydration synthesis to form long chains. There are 4 important polysaccharides that you need to know. The first is starch, which is the most important energy storage in plants. Starch is a combination of amylose and amylopectin. Amylose is a single chain and amylopectin is a branched chain. I have circled the individual glucose molecules within the chain for you to see. Cellulose, another important polysaccharide, a structural polysaccharide in plants. When you look at cellulose, it's 3 strands, long strands of glucose molecules with some special hydrogen bonds shown in between. Cellulose is what forms the important cell wall of your plants. Humans cannot digest cellulose, and it passes through our guts as fiber in our diet. Animals that have special enzymes released by bacteria called cellulase are able to metabolize cellulose and release the glucose. We do not get any energy from cellulose. In animals, our first energy storage is glycogen, which is a long polymer of glucose molecules. Here showing the actual glucose in your red circles. Glycogen is stored in the cells of the liver and the muscles, and so when we need to get sugar back, we need to go to our liver or muscles to release individual glucose molecules. This is actually showing glycogen, which is actually lots and lots of branched glycogen attached to a central protein that is stored in the liver or the muscles, mostly in the liver. The fourth structural polysaccharide is known as chitin. Chitin is an external polysaccharide that is found in fungal cell walls. It is also found in the exoskeleton of arthropods. It's a long polymer of N-acetylglucosamine, which is a derivative of glucose. It's the main component of the cell walls of fungi. It's found in the exoskeleton of some of your arthropods, crustaceans, crabs, lobster, shrimp, and even insects. And here this picture shows a cicada shedding its chitinous exoskeleton. And this is a picture showing the close-up of a sap beetle. This wing is also composed of chitin. So make sure you understand the difference between polysaccharides that are important in plants versus polysaccharides that you find in the animal kingdom.